Biological research requires scale and iteration. A single organism dividing exponentially provides the required mass to observe chemical processes in real time. Escherichia coli, specifically the K12 strain, is a gram-negative prokaryote. Through decades of rigorous observation, it has become the most thoroughly characterized biological system on the planet. Researchers do not observe E. coli passively. They deploy it as a highly tractable biological chassis, a stripped-down, programmable platform used to reverse engineer the mechanisms of life. While it exists naturally in the human gut, the specific strains cultivated in laboratories act as tightly controlled, calibrated instruments. To utilize this organism effectively, researchers must map its distinct biomechanical advantages directly against its inherent biological limitations. Deploying E. coli requires a strict accounting of its prokaryotic constraints. It is an engineered platform that only yields accurate data when operated within precise parameters. This split-screen diagram details the physical and genetic specifications that make E. coli the default choice for high-throughput research. On the left, an exponential growth curve plots its extremely short generation time. Under optimal laboratory conditions, E. coli doubles every 20 minutes. This rate allows scientists to quickly generate massive, statistically significant bacterial populations required for large-scale experimentation. On the right side of the diagram, we see its genome, a relatively small, fully sequenced structure consisting of approximately 4.6 million base pairs. Because this genetic sequence is entirely annotated, it functions as a complete, readable blueprint for basic cellular processes like transcription and translation. This minimal genetic architecture makes the organism highly manipulable. Scientists can alter its DNA with high precision to isolate specific gene functions and map regulatory networks. Culturing the bacteria requires standard, inexpensive media. This cost efficiency ensures that massive studies remain entirely reproducible across different global laboratories. The combination of rapid reproduction and an easily edited genetic sequence creates an optimal, high-throughput environment for foundational biological research. The utility of E. coli scales directly alongside the rise of molecular biology, starting in the mid-1940s. In 1946, researchers Joshua Lederberg and Edward Tatum discovered bacterial conjugation. They observed E. coli cells transferring genes through direct contact, establishing the organism as the primary model for genetic exchange. By 1952, the famous Hershey-Chase experiment utilized E. coli and infecting bacteriophages to definitively prove that DNA, rather than protein, was the true genetic material of life. Research then moved toward the chemical structure of genetics. In the 1960s, scientists entered a global race to decipher the genetic code itself. Scientists like Marshall Nierenberg and Heinrich Matthaei used E. coli extracts to map out exactly how specific sequences of DNA bases correspond to specific amino acids and proteins. These discoveries relied heavily on the E. coli cell acting as a perfectly controlled environment, allowing researchers to isolate deeply conserved biological mechanisms. This controlled environment also enabled the discovery of the lac operon, establishing the definitive model for understanding how genes regulate their own expression. These decades of E. coli research validated the central dogma of biology, mapping the standardized flow of genetic information from DNA to RNA to protein. In the 1970s, the methodology shifted. E. coli transitioned into the primary host organism for the new discipline of recombinant DNA technology. Researchers Herbert Loyer and Stanley Cohen developed molecular cloning techniques. They utilized restriction enzymes to cut DNA and plasmids to transport it. This schematic details transformation. Foreign DNA is inserted into an E. coli cell, forcing the bacteria to manufacture a specific recombinant protein. This process had immediate industrial applications, enabling the mass production of synthetic human insulin, growth hormones, and vaccines. This genetic flexibility drove continuous tool development. In 1987, Yoshizumi Ishino and his team discovered unusual repeated sequences directly within the E. coli genome, sequences later identified as the CRISPR-Cas system. 10 years later, in 1997, a project led by Frederick Blattner successfully sequenced the entire E. coli genome, cementing its status as an open-source biological tool. Today, research areas heavily reliant on this engine include metabolic engineering for advanced biofuels and high-throughput studies on the mechanisms of antibiotic resistance. The ability to directly edit E. coli at the genetic level positioned the organism as the primary engine of modern synthetic biology. The specific traits that make E. coli incredibly efficient, its simplicity and speed, also enforce strict experimental boundaries. This comparison highlights the primary limitation. E. coli is a prokaryote. It lacks the complex internal architecture, like mitochondria and a defined nucleus, found in higher organisms. Without these, E. coli cannot accurately model complex eukaryotic pathways or organelle functions. Because it is inherently single-celled, it provides zero insight into multicellular processes. It cannot model tissue differentiation, organ development, or the mechanics of cancer. It also faces metabolic limitations. E. coli operates on a simplified metabolic network that cannot perform advanced eukaryotic functions, specifically post-translational modifications like glycosylation. Furthermore, E. coli is prone to horizontal gene transfer. Bacteria can exchange genetic material directly, introducing unwanted variables that complicate experimental consistency. Environmental conditions present another barrier. E. coli thrives in environments distinct from human tissue, meaning data on stress responses or temperature sensitivity rarely translates. The evolutionary distance between a single-celled gut bacterium and a human means E. coli models usually cannot be applied directly to complex physiological systems. So how do researchers exploit E. coli's rapid iteration speed without being trapped by its prokaryotic limitations? They deploy complementary models. Pairing E. coli with yeast, C. elegans, or mammalian cell lines to study complex missing processes. Scientists also utilize advanced genetic engineering to humanize E. coli strains. They modify the bacteria to express specific eukaryotic pathways, allowing them to test complex functions in a simplified, controlled environment. When modifying the organism is impossible, researchers strictly limit their E. coli experiments to foundational, highly conserved biological processes, like DNA replication, that remain identical across all life forms. Working with these engineered or pathogenic strains requires strict adherence to biosafety protocols to prevent contamination and accidental release. By utilizing these strategies, modern research isolates specific human mechanisms within a bacterial vacuum, turning the organism's limitations into a controlled advantage. This chart positions E. coli among 10 primary models. Before committing, researchers process a strict matrix. First, does the study require eukaryotic metabolism or just conserved molecular processes? Second, does the design rely on rapid scalability? Third, does it demand direct clinical translatability? Researchers must also consider available infrastructure, utilizing comprehensive databases like Ecoli and the E. coli metabolome database to inform their study designs. Finally, utilizing a bacterial model offers distinct ethical and regulatory advantages, directly reducing the reliance on more complex, sentient organisms. Selecting E. coli is a calculated trade-off. Researchers explicitly exchange architectural complexity for unparalleled operational speed. E. coli functions as a specialized instrument for reductionist clarity, isolating core molecular processes away from the physiological noise of human systems. Decades of Nobel-winning experimentation have validated its position as the most well-characterized biological system on Earth. Its continued utility relies entirely on researchers acknowledging its rigid prokaryotic boundaries while fully exploiting its genetic tractability. By calculating those specific parameters, we continue to rely on a microscopic gut bacterium to program, produce, and engineer the future of life. The exact parameters of a discovery are always defined by the model chosen to test it. For foundational biology, E. coli remains the ultimate chassis.